A Scientific Look into the Causes of Epidermodysplasia verruciformis by Emily McCloy. What exactly is a wart? A wart is a small, hard, benign growth on the surface of the skin caused by the DNA containing the human papillomavirus, HPV. So how does this little bump just pop up? A virus enters the skin, often through a small cut or injury. When the virus is contracted, a wart does not appear or grow until about 1 to 8 months after incubation. Most warts are quite small and typically go unnoticed, but some can appear thick, large, and brown. These unsightly warts are nothing compared to the massive warts that have built up on the face and limbs of a 34-year-old Indonesian man named Didi Kaswara, also popularly known as the Tree Man. Didi suffers from a disease called epidermodysplasia verruciformis, further referred to as EV. This rare and invasive medical condition was first described in 1922 by the German dermatologist Felix Lewandowski and director of the Clinic Basel in Switzerland, along with his partner William Lutz. Felix and Lutz were credited for describing the disease, which can also be referred to as Lewandowski-Lutz dysplasia. So what in the world causes this disease that Didi suffers from? EV is an autosomal dominant inherited disorder that evolves progressively from early childhood through adolescence and into adulthood. Autosomal dominance is a pattern of inheritance that is characteristic of a genetic disease in which an individual has one copy of the mutant gene and one normal gene on a pair of autosomal chromosomes. Individuals with this disease have a 50-50 chance of passing the mutant gene to his or her children. This condition is universal and affects individuals of all races. The largest series reported includes 195 cases, mainly from Eastern Europe, Poland, and Latin America. Patients with EV typically present flat wart-like lesions on the extremities, face, and neck in early childhood, progressing into malignant tumors around 4 to 5 years old. Primary skin lesions manifest in two forms, although they are generally polymorphic, meaning they can appear in a variety of forms and shapes on the body. The first is a flat wart-like lesion that resembles verruca plana, a reddish-brown or flesh-colored flat-topped papule, which is a slightly raised pimple or swelling often found on the knees and elbows. The lesion is also commonly identified with scaly hyperpigmented or hypopigmented patches with irregular borders. Hyperpigmented patches are caused from the unusual darkening of the skin, and as you might guess, hypopigmented patches have an unusual lack of skin color, remaining very pale. Lesions may also closely resemble TNA versicolor, which is an eruption of brown patches on the skin caused by the fungus Pityrosporum furfur, often appearing white in contrast to hyperpigmented skin after sun exposure. The second form is seborrheic keratosis-like lesions, which are non-cancerous or benign wart-like growths only present on the surface of the epidermis that can be itchy and rough in texture and commonly visible on sun-exposed skin. A biopsy can be performed for early detection of premalignant and malignant lesions on patients with EV. The most characteristic finding of the condition is within the epidermis, where the flat wart-like lesions reside containing a mild hyperkeratosis, a thickening of the stratum corneum, which is the outermost layer of the skin, and within acanthosis, the deepest layer of the 5 layers of the epidermis. In this photomicrograph of a precancerous skin lesion from a patient with EV, the characteristic microscopic features of clear cells and in a large atypical nucleus in the center are visible. These features are commonly seen in HPV-associated epithelial cells. Here is another photomicrograph of the same skin lesion showing a positive staining of keratinocytes, the epidermal cells, infected with HPV type 8. The dark purple nuclear staining are the sites of the actual HPV DNA. A dense deposit of HPV DNA here has become visible with immunostaining, an antibody-based method to detect a specific protein in a sample. The cluster of blue dots is the presence of the infected DNA. The specific HPVs associated with EV can be divided into 2 groups. The first group has a high oncogenic potential, meaning the gene has the ability to become cancerous. 90% of patients who have this disease are associated with the HPV viruses 5 and 8. HPV type 5 infections infect cornified epithelium, a keratin formation or development of a horny layer of skin. and HPV type 8 poses a high risk for malignant conversions of skin lesions in patients. The lesions detected early on may transform into invasive squamous cell carcinomas, which is the cancer of epithelial cells that compose the main part of the epidermis. Squamous cell carcinoma accounts for 20% of cutaneous malignancies and 90% of all head and neck cancers. This photograph of invasive squamous cell carcinoma shows a warty lesion within sun-exposed skin of a middle-aged patient with ED. Chronic sun exposure coupled with common immunologic defects in patients are likely to induce mutations of the tumor suppressor protein p53 that is encoded by the TP53 gene located on chromosome 17 as well. A mutation in p53 leads to the development of malignant skin cancer in adult patients. The second group of HPVs, 14, 20, and 21, that are associated with this rare disease have a low oncogenic potential and are detected in non-cancerous benign skin lesions. The development of benign warts into malignant warts provides validity on defective cell-mediated immunity in patients with EV, where an immune response to the HPV infection does not involve antibodies, but rather involves the activation of phagocytes, the cells that protect our bodies by ingesting potentially harmful foreign particles and bacteria. The pathophysiology of EV linked to this immunologic defect provides elucidation on the pathogenic mutations found in the adjacent EVER1 and EVER2 genes, also referred to as TMC6 and TMC8, and located on chromosome 17. The role of these genes in the pathogenesis of EV remains unclear, but one hypothesis suggests that they are involved in the control of HPV infection with keratinocytes, the epidermal cells located on the outermost surface of the skin that produces keratin A fibrous proteins that form the main structural constituents of hair, feathers, claws, horns, and hoofs. Keratinocytes contain perinuclear halos, meaning situated around the nucleus, and a blue-gray pallor, which is a pale color on the epidermis that can indicate an illness is present. The transmembrane channel-like proteins 8 and 6 are integral membrane proteins encoded by the TMC8 gene and TMC6 genes, located on chromosome 17, and that is localized specifically to the endoplasmic reticulum, which is mainly composed of tubules and flattened sacs, responsible for many cell functions including protein modification and protein synthesis. Cancerous lesions containing high-risk HPV types 5 and 8 retain the integral protein E6 and E7, known to promote cellular growth, and which express portions of the viral genome. E6 and E7 regions work together to cause cell immortalization, resulting in the transformation of normal keratinocytes into malignant cells. With this information better understood, we can only begin to understand the difficulties Didi Kaswara has to live with every day. This rare and extreme case of EV inhibits Didi's ability to perform simple tasks such as eating, walking, and getting dressed. Didi was lucky to survive the extensive and painful surgeries performed to remove the thick mass of warts that resemble horns on his extremities and face. In 2008, doctors were able to remove 13 pounds of warts on his body. Skin grafts were used to cover freshly exposed skin previously hidden by the warts.
First, the surgery team must remove the massive cutaneous horns on his hand without amputating any of Dede's fingers.
Sangat sulit untuk mengidentifikasi tangan. Saya sangat sulit, jari.
Traditional surgery tools are too small to cut through 2 inches of solid growths. The surgeons realize a heavy-duty power saw is the only solution.
Kita sulit membedakan mana jari, mana horns. Sehingga nanti ketika kita buka, mungkin saja langsung tulang. Itu berbahaya sekali, bisa mati semua.
Each horn is taken off one at a time until the tops of the fingers are visible.
Jarinya juga ada yang
Dede's family watches helplessly.
Dia punya
Kuat tabah ya Pak ya.
When the saw breaks through, the skin must be cauterized to stop the bleeding. While cutting, the surgeons make a gruesome discovery. A beetle escapes. It's been living in Dede's horns.
95% of the warts were successfully removed with several surgeries, allowing Dede Koswara to move forward with his life and function more normally. Today, doctors are still searching for a cure to prevent his warts from growing back.